In this lecture, we are going to talk about the drug delivery systems based on nanoparticles. There are many kinds of nanoparticles have been applied as drug carriers nowadays. So let's begin the talk with the classification of nanoparticles. So nanoparticles used in drug delivery systems can be divided into categories like this. The ionic nanoparticles, lipid nanoparticles, and polymeric nanoparticles. So there are nanoparticles that is constructed from ionic bonding, from a kind of lipid compound like fatty acids and phospholipid, things like that. And some of nanoparticles can also be formed from polymers. First nanoparticles that I want to talk about is the ionic nanoparticles. So there are plenty of ionic compounds that can be used as ionic nanoparticles or ionic nanocarriers. But calcium phosphate is one of a very famous compound used widely. Because calcium phosphate composed of calcium ion and phosphate ion, and DNA and RNA also contains phosphate group in the structure as well, meaning that we can have such a strong interaction between calcium ion and the phosphate ion in the DNA. So if we mix the calcium ion together with the DNA, then with a strong interaction among them, we can then, after that, adding phosphate ion into the solution mixture. And then all of these will be co-precipitated as the nanoparticles with the controlled condition. So calcium phosphate nanoparticles embedded with DNA because of the strong ionic interactions. Calcium phosphate nanoparticles can be aggregated together very easy. So to prevent the aggregations and precipitate it out, we have to kind of doing kind of surface modification on the surface of our nanoparticles. So polymer like PEG can be applied onto the surface to prevent the aggregations by the steric effect of the polymer. So after we have this kind of nanoparticles, then we can treat the cells. So calcium phosphate nanoparticles can be get into the cells by endocytosis. So in the endosome, calcium phosphate can be disaggregated afterwards. So after this aggregation, the DNA can then infect it into the cells and doing their job. While in this case, they use the PEG alendronate. The alendronate is a kind of compound that can inhibit the mevalonate pathway, and inhibition of the mevalonate pathway can trigger the apoptosis, or the programmed cell death, right? So we can kill the cells, the cancer cells, by this kind of thing. Another very famous example is the calcium carbonate. So in this case, they want to treat the cancer cells with the etoposide, which is a non-polar drug. So the solubility in the water is not very good, and we want the selectivity to treat the cancer cells only. So first of all, the etoposide can be co-precipitated with the calcium carbonate, mix all of the components together, precipitate it out as a nanoparticles, and then inject into the blood vessel. Normally, the normal tissue will have a tight junction of blood vessel, but in the blood vessels that happen to be closer to the tumor tissue, the cell's junction is not very tight. So calcium carbonate nanoparticles can leak into the tissue. So the size of the nanoparticles should be something around 100-200 nanometer. So they can leak into the tissue and get into the cells by endocytosis as well. The tissue and the cells of the tumor normally has kind of acidic environment with a pH less than 6.5, less than 5, 5.5, 6.5, around that. So with acidic condition, as you know, the calcium carbonate or limestone can be digested easily. So after digestion, the drug etoposide can be released and then um, kill the cells, kill the tumor afterwards. So this is another kind of mechanism used. So calcium carbonate and calcium phosphate are very famous, and actually these ionic nanoparticles has kind of polymorphisms as well. So some structure of calcium carbonate and calcium phosphate has a high porosity. So if we can prepare the porous calcium carbonate nanoparticles or porous calcium phosphate or calcium apatite nanoparticles, then instead of co-precipitation, we can prepare the naked nanoparticles. So with the porosity here, we can just put the nanoparticles into the solution of the drugs. Then the molecules of drugs can get into the porous nanoparticle very easy. And apart from that, we can afterwards surface modify it with kind of receptors to give them the kind of selectivity properties. So in this case, in this study, they used dox or doxorubicin, a kind of drug for treating tumor, into the calcium carbonate nanoparticles, and with the surface modification of aptamer, then we can selectively put the nanoparticles into tumor cells. So with endocytosis in lysozyme, in the lysosome, the pH is less than 5, then calcium carbonate can be digested by the acidic environment, and then the doxorubicin is released. Next type of the nanocarrier is the lipid nanocarriers. The lipids like soap or phospholipid, sphingolipid, can be prepared into several different kind of structure. This depends on the bulkiness or shape and size of head group, which is very polar, and the tails, which is non-polar. So for example, if we have soap molecules with carboxylate as a head group and one hydrocarbon chain as a tail group, we can prepare the micelles. But if we're starting from phospholipids with phosphate as the head group and two tails of the hydrocarbon chain as the nonpolar part, right? So this is quite bulky, so we can constructing kind of lipid bilayer or vesicle out of the phospholipid. So we can constructing different kind of structure which are suitable for different kind of drug structure as well. Okay, first example of the lipid nanoparticles that I want to show you here is a kind of vesicle that is can be controlled releasing of the drugs inside. So the vesicle is constructed from the phospholipid. So as you can see here, our phospholipid composed of the phosphate group negative charge and the quaternary amine positive charge. This is the polar part. And we have two tails of hydrocarbon chain as a non-polar tail. And yeah, as a result, we can have kind of lipid bilayer. A very huge area of the lipid bilayer, they can curve up, constructing a kind of giant balls, which is called the vesicle. But anyway, this specific type of phospholipid, we embedded the azobenzene into the structure as well. This diazobenzene has kind of two geometrical conformation, cis and trans isomer. So cis and trans isomer, they have kind of different rigidity, right? So if we embedded the diazobenzene into the structure, we can switch cis and trans using like a kind of specific wavelength to switching from cis to trans isomer, and another suitable wavelength to turn it back from trans to cis isomer, something like that. So these we can then form a vesicle. Anyway, normally vesicle is very huge, and the aqueous environment in the center is bigger than this picture. Okay, but anyway, the aqueous environment in this picture, we can put polar kind of drug molecules into the structure. So for example, this is a glutathione. We can put the glutathione or any kind of polar drug inside, okay? And then we can release the drug out of the vesicle by using the advantage of our linker unit here. So now come to my questions. For students who take my course, you have to explain why we have to use the diazobenzene here as a linker, okay, and how we can control the releasing of the drug inside. Now I want to go to other examples. So what is the function of the linker? So just the question that you have to answer me. Next type is a solid lipid nanoparticles. So previously we have the polar drug that we want to encapsulate it, right? So we put into the aqueous environment of the vesicle. Now if the drug is nonpolar, so what we have to do is constructing other kind of lipid, which is a solid lipid nanoparticles. This structure is very similar to the kind of lipoprotein, but lack of the protein. Right? So lipoprotein constructing from the phospholipid and proteins, and in the center there is nonpolar. So lipoprotein is for transporting the triglyceride cholesterol, something nonpolar, into our blood vessels. Okay. But now, if you want to transport the nonpolar drug, huge amount of nonpolar drug, right? So we need a very huge area of nonpolar environment. So solid lipid nanoparticle is the answer. So first of all, we have to use the drugs and then mix with the natural lipid like triglyceride, like fatty acids, and also the cholesterol. Right? So all of them, mix them up together, and then we can trap it into the emulsifier or into the phospholipid. Right? So at the end we have solid lipid nanoparticles, which is very similar to the lipoprotein, but without the protein on the surface. So this solid lipid lipoprotein can use by oral administration, eat them up, right? And then they get into the circulation into our body afterwards. So this is for nonpolar drug. Another thing is the polymeric micelles. Polymeric micelles, instead of using kind of phospholipids, which is quite expensive actually, phospholipids, if you go into the chemical catalogs, you can see the price of the phospholipid. All of them are very expensive. So instead of using phospholipids, so some using synthetic polymer instead, but the polymer here have to be biodegradable polymer to avoid problems with our body, right? So we have to use the kind of biodegradable polymer. So in this case, if we want to transport the doxorubicin over here, which is very nonpolar as well, so doxorubicin have to be trapped inside the polymeric micelles. So in this case, we have diblock copolymer in this picture. So we can actually prepare diblock copolymer or triblock copolymer. So as you can see here in the structure, the left-hand side blue color is PEG, polyethylene glycol, which is very polar. Right? And then on the right hand side in pink color is non-polar polymer, right? Because this is the polyester with 6 carbon in the structure in the repeating unit. So this is quite non-polar. So this kind of polymer can act as similar to the soap molecules, right? So the pink color turn themselves together and then stack together by the London force or hydrophobic interactions, while the blue color dissolves well into water, they point it outwards. So doxorubicin can be trapped inside the structure of the micelles in the pink region of the polymer. So this is diblock coat polymer. So we trap the doxorubicin in the center, and then we can constructing the micelles and transport it into the aqueous media, because doxorubicin cannot dissolve well in water, so we have to use kind of polymer to like to be as a carrier. Another advantage is we can prevent the doxorubicin from dissolve into the blood vessels, go to any organs, but we can control the flows of our doxorubicin. And moreover, sometimes we can also doing a surface modification by chemical reactions with the blue parts, right? So we can then selectively direct the nanoparticles to some kind of cells, some kind of cancer cells, using the receptor or antigen antibody interactions, things like that. So these are kind of examples of nanocarriers. Okay. So for my students, do not forget to submit the answer of my questions. Okay, it's due in the next week. Okay. So anyway, this is just roughly introduction of the nanoparticles that can be applied as a nanocarrier. But okay, thank you very much for your attention. I hope you enjoy the examples and the chemical structures over here. Thank you very much, and see you next time.